Hello class, welcome to our Biochemistry Laboratory post-lab discussion. For today, we will be discussing the topics under your breaking proteins and a little bit from your laboratory safety. So eliminate na natin yung topic under osmosis, which is the bouncy egg, kasi na-discuss na natin siya for period 1. So you have to make notes. Mag-take down notes po tayo as we will be using it for the laboratory examination later on. But first, let us discuss these minor changes that you will be observing on your period 2. For section 1 hanggang section 4, meron lamang tayong slight changes with regards to the laboratory instructors who will handle your class as well as the posting of the activities on FinMag Google Classroom. For section 1, same pa rin naman, it will be handled by Ma'am Trina and Ma'am Trina pa rin po for laboratory. Like and lab, wala namang changes sa schedule. For number 2, wala namang changes sa schedule of the lecture and laboratory part. However, sa laboratory, it will be handled by Ma'am Snowy. Sa lecture part, Ma'am Trina pa rin naman po. Pakitandaan po at pakigawan na po ng screenshot. For section 3, yung schedule, meron lang na bago sa laboratory part. Sa lecture, same pa rin naman. It will be handled by me. And for the laboratory, it will be moved from 7:30 to 11 a.m. on a Saturday basis and will be handled by Ma'am Lena and Ma'am Nina. Lastly, the section 4, ang lecture part same pa din, will be handled by me, but the laboratory part will be moved on a Saturday basis and will be handled by Ma'am Lena and Ma'am Nina. So ma'am, ano po ba yung mga changes na mangyayari kapag nagbago na yung instructors namin, as well as bakit pa po ba binigay sa kanila. Okay? Kung napapansin nyo, yung biochemistry and molecular biology, ako yung nag-handle nun. So, sobrang daming load. Hindi nakakayanin ni Ma'am Trina. Okay? And then, nandito po yung ating mga other instructors, other professors to help me. Okay? And to help us, help me guide you guys. And then, what else? Meron po tayong frequently asked questions na sasagutin natin on our next slides. Okay, so tandaan niyo na po yung mga laboratory instructors nyo. Next, yung nasa FinMag Google Class po ba ng mga experiment or SAS module, need pa po bang ulitin or mark done na lang? So kung magpo-post kami sa FinMag Google Classroom, for sure, mark done na lang yon. Pakiantay po yung instructions coming from your professors on what to do on such activities. But most likely, mark done na lang yon. Kasi nga nagkaroon tayo ng mga discrepancies sa ating FinMag Google Classroom. So, we switched to old Google Classroom muna. But most likely, mark done na lang yan. As you can notice, si Ma'am Trina hindi pa po nagpo-post sa biochemistry or molecular laboratory na mga experiments or other activities. Kung meron man, hindi po ako yun. Hindi ko pa kasi na-check talaga siya. Okay, mark down nyo na lang kasi na-submit nyo naman na yun sa old Google Classroom. Question number 2, kanino po magka-clarify ng grades sa experiment or any lab activity? So, balikan natin yung slide natin kanina. Section 2, for example, meron kayong grade sa isang experiment and meron kayong gustong i-clarify kung bakit 40 over 50 lang yung score nyo. Okay, that will be the time na i-approach nyo si Ma'am Snowy. Okay, kasi siya na yung mag-grade, kasi siya na yung mag-handle. Okay, for section 3 naman, bakit po Ma'am 0 yung binigay niyo sa akin and nag-submit naman ako on time? Pwede niyo na pong i-clarify yon kay Ma'am Lena or kay Ma'am Nina. Okay po? Pero pagka meron namang pinost na activity, let's say si Ma'am Trina yung nag-post, makikita niyo naman yun kung sino yung module writer. O kaya marinig niyo yung boses na nagbibigay ng instruction. Nag-post si Ma'am Trina. Ang sabi, ito yung enzymatic activity na gagawin natin for period 2. Tapos hindi niyo maintindihan kung or ano pong pwedeng alternative doon sa experiment natin, ma'am. Pwede po ba kami gumamit ng ganito? Pwede niyo pong itanong kay Ma'am Trina yon as module writer, for example, or pwede niyo itanong kay Ma'am Snowy kasi for sure alam nila yon. Kasi aaralin din naman nila yung experiment na yun. Kasi aaralin niyo yun, aaralin din ang mga professors niyo yun. So yun po yung clarification natin. With regards doon sa grade ninyo, sa experiment or sa mga module, doon na po kayo sa instructor mismo per section na naka-assign lalapit. Pero kung yung regarding doon sa clarifications ng inyong experiment or any activity, you may approach any one of us. Next, kanino po magka-clarify regarding sa module posted? Yun na nga po, nabanggit ko na kailan po gagamitin yung Finma Google Classroom. Ako, Ma'am Trina, starting period 2 na tayo lahat. Kasi na-encounter natin yun last semester na hindi lahat nagte-turn in. Diba? 5 minutes lang naman mag-turn in pero hindi po lahat nagko-comply. So ako, para walang problema, walang markdown, P2 na ako. Starting module 11 doon na tayo, magmamigrate na tayo. Okay, pasensya na kung medyo nakakalito, ang daming pinapagawa, ang daming pinapakomply. Hindi rin naman po namin kagustuhan. It's just that magdagkaroon lang ng mga discrepancies sa Finma Google Classroom. Pakiantabayanan po parati yung instructions ng inyong mga professors kasi iba-iba kami ng instructions regarding sa Finma Google Classroom. But for Ma'am Trina, yung 2 major subjects yung molecular biology and biochemistry, hindi pa po kailangan mag-mark down. Meron man na post, i-mark down nyo na lang. Anyway, kahit hindi kayo nagpasa, makikita naman ng prof, ibabalik rin naman yan sa inyo para wala lang nakapending dun sa activities ninyo. Number 5, ma'am, bakit pa iba-iba naguguluhan na po kami? Yes, kami rin po naguguluhan, mag-comply na lang po tayo and then magtulungan tayo. Okay po ba tayo doon? Number 6, ma'am, bakit ang daming dates ng lab exam? Bakit di na lang po sumunod according sa schedule? Nagbigay ako ng different schedules ng inyong laboratory exam para makita kung ano yung best fitted dun sa inyong schedule. Kasi marami tayong mga working students as well as may mga emergencies na nangyayari na hindi naman natin ginusto. So, para ma-avoid na natin yung special quiz, and then para ma-avoid na natin yung mga hindi makapag-take ng quiz, meron tayong iba't ibang schedule ng laboratory exam natin. Pili na lang kayo kung ano yung best fitted sa schedule nyo. Pasensya na, medyo magulo si Ma'am Trina, nag-a-announce bigla-bigla ng ganito kasi bigla rin akong may mga ina-attendan na emergency meeting. And lastly, mag-e-exam po ba kami lahat, ma'am, sa P1 lecture? May permit or wala? Yes po. Nabanggit natin to sa ating orientation video or orientation natin na mag-e-exam kayong lahat kasi 100 items. Mahirap na matambak siya lahat doon sa dulo. Kasi anyway, lahat naman din kayo magbabayad sa dulo ng ating semester. So ganun din naman, papag-takein ko na kayo lahat. So kung meron man akong instructions kung paano mag-take yung mga walang permit, kung paano mag-take yung may permit, pakiabantay na lang po, pakiantabayanan na lang po. Basta ang mahalaga, lahat kayo magtitik. So let us just have our post-laboratory discussion regarding your laboratory safety. We will begin with the chemical hazard identification system. You have to know this by heart as this will appear on your laboratory examination as well as the board examination. Pakitandaan si yellow, white, blue, and red, you were born right. Nakikita po talaga ito sa isang totoong laboratory setting. Yes, itong mga picture na to, itong yellow na box dikit po talaga siya, posted siya sa mga cabinet kung saan nakalagay ang inyong mga chemicals. Yellow means instability, the white is for your specific hazard, the blue stands for the health hazard and then is categorized from 4, 3, 2, 1, and 0. And lastly, your fire hazard that has flash points from 4, 3, 2, 1, and your 0. Pakitandaan na po yan. Next, these are the common signages that you will observe when working inside the laboratory. Yung most common dyan si radioactive, si biohazard, as well as the poison. However, lahat ito maaari natin ma-observe sa ating laboratory. Next, for your breaking proteins, we were able to do an experimentation regarding the proteins na meron tayong 4 cups on which naglagay tayo ng albumin or our egg and then sinubject natin siya sa iba't ibang chemical or physical changes. Number 1 is the control. Egg whites start out clear. They are almost 90% water, but the other 10% is packed with proteins. Egg white contains more than 50% of the proteins found in the egg. Sa control, wala tayong ginawang intervention or anything. nag-stand lang siya doon, it serves as a standard or a control. Control po talaga siya. And then the main protein in the egg is what we call the albumin, the small folded proteins in the egg. Egg white don't take much space. The gel-like egg white looks clear. The control egg showed us that when left at the room temperature, the egg white stays clear, meaning the proteins maintaining their original shape, and then these proteins were not denatured. So ano po bang sabi natin kapag wala tayong ginawang interventions or changes sa ating control, wala rin pong mangyayaring changes na makikita or ma-observe ninyo doon sa ating egg white or doon sa ating albumin. So ano pong nangyari na hindi na denature yung protein? Ibig sabihin ng denaturation or the denatured, hindi nag-break yung linkages inside the protein, inside the amino acid or what we call your peptides. Kasi yung mga peptide bonds natin stable yan. And then yung protein, meron siyang mini-maintain na original shape. So once na sinubject natin ang ating mga albumin or ang ating mga proteins into slight or minor changes, nagkakaroon din ng changes sa original shape kasi nga nagbe-break yung mga linkages doon sa kanyang mga peptide bonds. Next, when we subjected our egg or albumin into a hot water, it is whenever eggs are cooked with heat, the egg whites turn from clear to white and the gel becomes rubberier. And then as heat denatured the proteins in the egg white, it broke apart some of the bonds, mostly your hydrogen bonds that were holding the proteins in their original shape. And then the proteins unfolded, taking up more space, turning into gel white and hardening them in the place next to one another. It is when your proteins are denatured. Sa hot water, nung nagkaroon ng slight changes sa kanyang temperature, ang nangyari, nag-denature ang inyong protein. So kung i-recall nyo yung experimentation nyo, nagkaroon ng changes doon sa inyong egg white. Your denaturation by heat can be used to disrupt your hydrogen bonds and non-polar hydrophobic interactions. This occurs because heat increases the kinetic energy and causes the molecules to vibrate so rapidly and violently that the bonds are disrupted. Kapag sinubject natin ng ating protein into gentle heating, it causes reversible denaturation of protein. But when we subject our protein into vigorous heating, it can cause irreversible denaturation of the protein. Heat coagulates and destroys protein present in the bacteria. Thus, sterilization of instruments used in medical setting requires use of high temperatures. Yes, totoo po na ang heat, it can kill the bacteria, targeting the proteins in the bacteria. However, meron tayong specific temperature na kailangan i-reach para mamatay po yung mga ganong pathogens. Okay, hindi lang basta napakuluan siya. Meron tayong ni-reach na certain temperature. Next, after naman natin yung inyong heat, yung egg na may heat, yung egg na nasa control, ito naman si egg na may alcohol. Alcohol denatures proteins. It does the same way by heat by breaking the bonds that holds the parts of the protein in a folded shape. Sometimes the alcohol molecules bond directly to some of the parts of the protein, disrupting the normal way the protein would bond to itself. So the alcohol is called bond disruptor. The proteins again unfolded, taking up more space and hardening in place to next one another. This process took much longer with alcohol than it did with heat. However, the longer the time for denaturation with alcohol is simply because it spreads more slowly than heat. The alcohol had to diffuse or move through fluid into the egg in order to affect the proteins it touched. Yes, the alcohol denatures your proteins. However, yung heat ang mas mabilis mag-akapan doon sa inyong mga proteins. Kasi nga po, meron tayong kinetic energy coming from the heat. Okay, remember yung inyong gas, yung inyong heat is coming from the gas. So, mas mabilis yung movement ng molecules compared doon sa inyong liquid. Pareho naman nilang na-disrupt yung hydrogen bonds of the protein. However, mas mabilis lang yung nasa gas, yung nasa heat. Kasi nga, mas freely moving yung mga molecules ng inyong gases. Okay, so Chemistry 101. Next, when we subjected our egg into room temperature water, sometimes in this experiment, room temperature water has a small denaturating effect on some of the egg white. It acts the same way by breaking bonds, but its effect isn't nearly as strong as alcohol or your hot water. And then these are the other ways to denature your protein. It can be meant by coagulation that has your heat, alcohol, and inorganic acids such as your hydrochloric acid and your sulfuric acid. And then by precipitation, by means of heavy metal salts and alkaloidal reagents. So ano nga po ba yung nangyayari sa precipitation? The protein will precipitate because the ionic bonds and hydrogen bonds are disrupted. So yun ang parating goal ng inyong mga denaturating reagents. That is to break the bonds or disrupt the bonds or hydrogen bonds and ionic bonds of your proteins. And then, in cases of heavy metal poisoning, egg whites can be used as antidotes. Denature proteins irreversibly by disrupting the salt bridges and the disulfide bonds present in the protein. In a situation, a person may have swallowed a significant quantity of heavy metal salt. An antidote may be administered to precipitate the poisonous salt. Then, an emetic can be given to induce vomiting so that the precipitated protein can be discharged from the body. So yun, kapag meron kayong na-experience na ingest ng large quantity or large amount of heavy metal poisoning, na poison siya doon, egg whites can be used as antidotes. Then, pwede nyo po siyang bigyan ng mga emetic reagent or emetic medicine para ma-induce yung vomiting at maisukan niya yung heavy metal toxins kasabay noong egg white. Okay, parang nano-neutralize lang niya yung effect ng heavy metal poisons nyo. And then these are the alkaloidal reagents. We have your picric acid and then the tannic acid. And then for your protein precipitation, these are the actual experimentation that we will be doing when we return to face-to-face classes. Ito na talaga yung gagawin yung experimentation. Kasi yung na-discuss natin kanina, tungkol yun sa ating what we call laboratory experiment at home. So ito na yung actual talaga. So pakitake note na lang po. The precipitation by strong inorganic acid. Ma'am, kanina pa po natin pinag-uusapan si precipitation. Ano nga ba yung precipitation or precipitate? Yung precipitate, naalala nyo yung parang mga anong tawag doon? Yung mga maliliit na particles inside your solution. Your precipitate are what we call the insoluble solid that emerge from a liquid solution. Yung mga precipitate, in Tagalog, yung mga latak-latak ba? Yun, in Tagalog siya. But in science, speaking, it is the insoluble solid that emerge from a liquid solution. So this is what we call the Heller's test. It is a biochemical test performed to detect proteins in a sample by denaturation of proteins by the addition of your strong acids. The principle behind this test is that this method relies on the changes of a solution pH. Every protein has a defined isoelectric point or TI value. Little changes in a medium pH have an impact on the protein structure. Adding acids to the solution lowers the pH and leads to positively charging the protein due to the proton captured by your amino acid. So kung matatandaan, mag-introduce man tayo ng slight changes sa ating mga proteins, it will surely affect the structure of that specific protein. Next, your concentrated inorganic acid. Example, nasa laboratory tayo and then gagawa tayo ng isang experimentation. We have your test tube number 1, test tube number 2, and test tube number 3. And then lagyan natin sila ng, for example, urine ng kaklase ninyo. Or for example, albumin na lang. Okay? And then, mag-start muna tayo sa urine. Lagyan mo muna siya ng concentrated hydrochloric acid, sulfuric acid, and nitric acid. So sa Heller's ring test, pagka nilagyan mo ng urine ng kaklase mo, for example, and then lagyan natin ng concentrated reagent, dapat kung walang presence ng urine doon sa test tube ninyo, dapat wala pong mapo-form na precipitate doon sa taas. Okay, so ang albumin, dapat po bang nakikita yan sa urine? Dapat po bang present siya? Yes po, tama po ang sagot, hindi po, not at all. Kasi sa laki ng mga albumin na yan, dapat na-filter na yan ng ating kidneys and it does not reflect or you won't observe any albumin on your urine. Dapat po wala. So for example naman, gawin natin yung tatlong test tube and 1 ml each contains 1% filtered albumin solution. So since dito known na may filtered albumin solution ka, it will surely form a precipitate doon sa taas. Pero kung ang gagamitin natin urine ng kaklase ninyo, dapat po wala po tayong makikitang precipitate doon sa taas. Kahit gaano karami pang reagent ang ilagay natin. So ito yung ating tabular form of your concentrated hydrochloric solution. Kapag meron tayong known na albumin, it will surely form a white precipitate. Kahit mag-add ka pa ng excess reagent, white precipitate pa rin yan. Pag meron naman tayong concentrated sulfuric acid, pag nag-add tayo ng known 1% ml of the albumin, it will form a pinkish precipitate. Pag dinagdagan natin yung ating reagent, it will form 2 layers of white precipitate. And lastly, your concentrated nitric acid, pag meron tayong kasamang 1% ml of your known albumin, it will form white precipitate. But upon addition of your excess reagent, which is the nitric acid, it will form yellow precipitate doon sa taas niya. Next, the precipitation by alcohol. Its principle is that alcohol or your organic solvent precipitates a proteins by removing water shell around protein molecule, which is your dehydration, leading to denaturation. Hydrogen bonding occurs when amide groups in secondary protein structure. Hydrogen bonding between side chains occurs in tertiary protein structure in a variety of amino acid combinations. All of these are disrupted by the addition of your another alcohol. So lahat sila gustong i-disrupt sa protein structure. So kung gagawin natin siya sa isang laboratory setup, ganito po siya. Maglagay tayo ng 1 ml ng albumin, gelatin, and peptone. And then meron tayong isa-subject sa under 95% alcohol per test tube. Ma-observe natin na wala po tayong coagulum na form. Next naman, yung i-repeat natin yung itong step na to, for example, and then gumamit tayo ng 70% alcohol, makaka-observe tayo ng precipitate. Merong formation of white precipitate. So, ganito po siya. So, 95% alcohol, di ba? Ito naman sa 70%, and then ito po siya. So, sa protein precipitation by alcohol, kapag gumamit ka ng 95% alcohol, sa 1% albumin, yes, may coagulation. Sa 70% alcohol, may light precipitation but clear solution. Okay? Ito po siya. Sa 1% gelatin, you will observe a defined coagulation, but in 70% alcohol, light precipitation lang siya. And then for your 1% peptone, it will have a 95% alcohol and then it will form a coagulation. But for 70% alcohol, light precipitation lang siya. Ito po yung itsura. And then the precipitation by heat coagulation test is used to determine the presence of albumin and globulin protein in the urine sample, as albumin and globulin in urine are observed under many pathological conditions. Their presence can be conclusively established by these tests, which aids in the diagnosis of your disease. So sa ating laboratory setup, sa clinical microscopy particularly, marami na tayong innovations na nagawa doon. Meron tayong urine strips and machines that are able to read the microscopic contents of your urine. But if you will go back to the basic, ganito po natin siya babasahin, yung mga precipitation by heat and then the protein test and the likes. Okay? And then your precipitation by heat its principle is the heat disrupts your hydrogen bonds of the secondary and tertiary protein structure while the primary structure remains unaffected. The protein increases in size due to denaturation and coagulation occurs. So yung coagulum formation niya, nade-disrupt yung inyong protein. So kaya nagkakaroon po ng parang turbid na itsura. So pag sinubject naman natin ng ating test tubes na gagamitin natin for experimentation through heat, meron tayong test tube number 1, 2, and 3. This is what we call yung burette. Burette test. Okay? Ito po siya. So yung test tube 1, 2, and 3 nyo, basta pakitandaan po pag positive at may presence ng protein, it will form a violet filtrate. Magtuturn siya into a violet. But pag wala naman pong presence ng inyong protein, and then if you will precipitate it by heat, okay, it will turn into a blue filtrate. Pakitandaan po yan for your burette test. And then these are the questions that we are commonly asked. For the denaturation and the degradation, what is the difference between these two? Protein denaturation is a process modifying the molecular structure of your protein. Also, your denaturation involves the unfolding of a protein where quaternary, tertiary, and secondary structures are disrupted, but your primary structure remains intact. Meanwhile, for your degradation, in protein degradation, the primary structure is destroyed, which means the covalent bonds are broken. Covalent peptide bonds are broken. And then this means that the covalent bonds between amino acid is broken. This can typically happen after the treatment with certain enzymes called your proteases. Well, that concludes our post-lab discussion for period 1. Thank you and good luck to your examination later on.